Here are 5 high-yield USMLE-style questions focused on microtubules, their structure, functions, and related clinical applications. Question 1. Which of the following drugs works by disrupting microtubule polymerization and is commonly used in the treatment of gout? A. Mebendazole, B. Griseofulvin, C. Colchicine, D. Vinca alkaloids, E. Taxanes. Answer C. Colchicine. Explanation. Colchicine works by disrupting microtubule polymerization, inhibiting inflammation in gout by preventing neutrophil migration and activity. It is specifically used in the treatment of acute gout attacks. Other drugs, such as mebendazole, griseofulvin, and vinca alkaloids, also target microtubules, but for different conditions: antihelminthic, antifungal, and anticancer, respectively. Question 2. Which of the following best describes the role of dynein in microtubule function? A. Transporting cellular cargo toward the positive end of the microtubule. B. Moving cellular cargo toward the negative end of the microtubule. C. Forming the structure of mitotic spindles. D. Synthesizing tubulin dimers for microtubule assembly. E. Catalyzing the hydrolysis of GTP during microtubule polymerization. Answer B. Moving cellular cargo toward the negative end of the microtubule. Explanation. Dynein is a molecular motor protein responsible for retrograde transport, meaning it moves cellular cargo toward the negative end of the microtubule, which is typically toward the nucleus. In contrast, kinesin is responsible for anterograde transport toward the positive end, toward the periphery. These motor proteins are crucial for the intracellular transport of organelles, vesicles, and other cellular materials. Question 3. A 45-year-old male with a history of herpes simplex virus (HSV) infection presents with painful lesions on his lips. Upon examination, you notice the reactivation of the virus. Which of the following molecular motor proteins is primarily responsible for transporting the virus from the neuronal cell body to the periphery? A. Dynein, B. Kinesin, C. Myosin, D. Actin, E. Vimentin. Answer. Explanation. In HSV reactivation, the virus uses anterograde transport from the neuronal cell body to the periphery via the motor protein kinesin, which moves along the microtubules toward the positive end of the microtubule, which is directed toward the periphery of the cell. Dynein, on the other hand, is involved in retrograde transport. Question 4. A 4-year-old child is diagnosed with a helminthic infection. The physician prescribes a drug that inhibits microtubule polymerization to treat the infection. Which of the following drugs is most likely prescribed for this condition? A. Mebendazole, B. Vinca alkaloids, C. Taxanes, griseofulvin, E. Colchicine. Answer A. Explanation. Mebendazole is an antihelminthic drug that inhibits microtubule polymerization in helminths, parasitic worms. This action prevents the formation of essential structures such as microtubules in the parasite, leading to its death. The other drugs listed are either antifungal, griseofulvin, anticancer, vinca alkaloids, taxanes, or anti-gout, colchicine. Question 5. A 50-year-old male with a history of cancer receives taxane therapy as part of his treatment regimen. Which of the following best describes the mechanism of action of taxanes? A. Inhibition of microtubule polymerization. B. Inhibition of microtubule depolymerization. C. Inhibition of microtubule assembly. D. Disruption of actin filaments. E. Inhibition of DNA synthesis. Answer B. Inhibition of microtubule depolymerization. Explanation. Taxanes, e.g. paclitaxel, are anti-cancer drugs that inhibit microtubule depolymerization, effectively stabilizing the microtubules and preventing their disassembly during mitosis. This disruption of the normal dynamics of microtubules impairs cell division, leading to cell cycle arrest and apoptosis, particularly in rapidly dividing cancer cells. These questions test your MLE-style question that includes the mnemonic. Question. A 45-year-old male is diagnosed with gout and is prescribed colchicine for management. The drug works by disrupting microtubule polymerization and inhibiting neutrophil migration to sites of inflammation. Which of the following drugs also acts on microtubules, but has a different mechanism of action by stabilizing microtubules and preventing their depolymerization? A. Mebendazole, B. Griseofulvin, C. Vinca alkaloids, D. Taxanes, E. Colchicine. Explanation. Taxane, e.g. paclitaxel, stabilize microtubules and prevent their disassembly, which is the opposite of how colchicine works by disrupting microtubule polymerization. Both classes of drugs affect microtubule dynamics, but they do so in contrasting ways. A helpful mnemonic to remember these drugs and their actions is mice get colder very tired, which stands for M. mebendazole, antihelminthic, G. griseofulvin, antifungal, C. colchicine, anti-gout, V. vinca alkaloids, anti-cancer, T. taxane, anti-cancer. Vinca alkaloids inhibit microtubule polymerization, while taxanes stabilize microtubules, highlighting the difference in their mechanisms.